Hello friends, welcome back to my channel, and today in this video we are going to study about primary lymphoid organs. So primary lymphoid organs means these are the organs that helps in the development of lymphoid cells. So under this the first organ is bone marrow, which is a kind of primary lymphoid organs that supports self-renewal and differentiation of hematopoietic stem cell into mature blood cells. Although all bones contain marrow, but the long bones femur is our thigh bone, humerus our arm bone, hip bones ilium, and sternum is the bone which is present in the middle of our rib bone. The central bone connecting the side rib bones, that is known as sternum. So these are the sites of most active hematopoiesis, but bone marrow means it is present in all the bones. But coming to these long bones, hip bones and sternum, they are the most active site of hematopoiesis. Hematopoiesis means the process that develops hematopoietic stem cells, self-renewal of hematopoietic stem cell, and also differentiation of various other blood cells, which include lymphoid lineage, myeloid lineage. Under lymphoid lineage, we have T cells, B cells, NK cells. Under myeloid lineage, we have macrophages, erythroid cells, dendritic cells found in both the lineages. This we already learned. Now, if we see the structure of an adult bone marrow, it contains cells that help in the development of hematopoietic stem cells. HSC means hematopoietic stem cell. Now, hematopoietic stem cell is present in bone marrow, but bone marrow contains, along with these hematopoietic stem cell, other cells that helps in the development and differentiation of these hematopoietic stem cells. These are osteoblasts. Osteo means bone cells. They generate bone and control the differentiation of hematopoietic stem cell. Endothelial cells. These cells line the blood vessels and regulate HSC differentiation. Reticular cells, they connect cells to bone and blood vessels. Sympathetic neurons, they control the release of hematopoietic stem cell from bone marrow. Depending upon the signal, they will release the hematopoietic stem cell. Okay. Now, if you see the diagram, here this is hematopoietic stem cell. Apart from hematopoietic stem cell, here we have osteoblast cells also. And this region is known as endosteal niche. Apart from this, here in the center, we have arteries and veins. That means this is vascular niche. Now, the hematopoietic stem cell, as you can see the arrow here, coming towards the center. So, this is hematopoietic stem cell, the undifferentiated hematopoietic stem cell. When this stem cell will proceed in the path of differentiation. So, along this path of differentiation, it will move from this endosteal niche towards vascular niche, that means towards the center, because here only the arteries and veins are connected. That is the blood vessels that helps in supply of blood towards or away from the bone marrow. So the undifferentiated stem cells, they are present towards the outside, towards the border of the bone marrow. As soon as they become differentiated or they progress in the path of differentiation, they'll come towards the center of the bone marrow, which is known as vascular niche, because in the center only the arteries and veins are connected. So from here only the differentiated hematopoietic stem cell, that means T cells, B cells, NK cell, erythrocytes, this will enter in our blood vessels and will get supply in our whole body. So always the undifferentiated cells are present on the boundaries and the differentiated cells are present in the center, because from the center only the arteries and veins are connected. Now with the age, this efficiency of hematopoiesis decreases. Endosteal niche contains quiescent HSC, that means undifferentiated one, and the vascular niche contains differentiated hematopoietic stem cell. Now coming to thymus, it is present just above the heart and the site of maturation of T cells. The immature T cells that are called thymocytes, that means we are talking about nerve cells only. Now, thymus is divided into cortex, the outer cortex, and inner medulla. The outer cortex consists of DP cells and the inner medulla consists of SP cells. DP means double positive, SP means single positive. We'll understand what are these. The extreme boundary is called as capsule, the gap between capsule and cortex, subcapsular cortex, and then we have cortex and inner medulla. And we have a junction called cortical medullary junction. This is the junction from where the blood vessels can enter and exit. Now, what do we mean by DN, DP, and SP? Now, we are talking about the maturation of T cells. T cells, we already learned, divided into two broad categories, TH and TC cells. How we differentiate on the basis of markers? These are CD4 plus, these are CD8 plus. So the naive T cells are first in the form of DN. DN means double negative. Double negative means CD4 negative, CD8 negative. That means they do not contain any markers, neither CD4 nor CD8. These are known as DN cell. These DN cell will enter the cortex. When they enter the cortex, they become DP. DP means double positive. That means now they contain CD4 plus and CD8 plus, both the markers on the surface. Towards medullary or medulla, when they enter inside the medulla, now they are SP, that is single positive. That means either CD4 plus or CD8 plus. This is the stage when the T cell get differentiated into TH and TC cell. Till cortex, they are only T cells. We cannot say them as TH or TC cells because either they are double negative or they are double positive. So we cannot say them as TH or TC cell. We call them as a T cells only. Once they are in medulla, now they are SP, single positive. So they contain either CD4 plus, that means now we can call them as TH cell, or they contain CD8 plus, that means now we can call them as TC cells, because we know that TH contains CD4 marker and TC contains CD8 marker. So now we can differentiate them or call them as TH and TC cell. Now, how this happen? Why the cells are double negative in the beginning? How they become double positive? And again, how they became single positive. So this we'll understand in the next slide. So let's see what is happening here. When the cell is in bone marrow, thymus is the site of maturation, but all the cells are formed or developed in the bone marrow only. Coming to B cells and other cells, they mature inside bone marrow. But coming to T cells, after developed in the bone marrow, they will migrate towards thymus and undergo maturation. So in the bone marrow, they are thymocyte. Thymocyte means C minus minus. That means double negative. No CD4, no CD8. Now when they are inside the thymus, but in the cortex of the thymus, now they will undergo positive selection. We learned about positive and negative selection. What is the difference? Suppose we have two types of cells, this colored one and these hollow ones. For our work, we need these hollow ones. So we have two methods of selecting the hollow ones. Either we can directly select hollow ones in a tube. So this is positive selection. We are directly picking and selecting the hollow ones. The second method is we are selecting the colored ones and then discarding it. So we will left with only hollow ones. So this is negative selection. Positive means we are selecting the hollow ones. Negative means we are selecting the colored ones and discarding it. So we are now left with only the hollow ones. So this is a method of negative selection. So now coming to the cortex of thymus. Here what is happening? Positive selection. How it is happening positive selection? The T cell is double negative now. In the cortex of the thymus, they'll get the opportunity to bind with MHC, the cells that contain MHC marker. Now, because these are T cells, they should gain the ability or they should have the property to bind with MHC, either class 1 or class 2. They should bind with the MHC because T cell recognize antigen only when presented via MHC. This we already learned in the last videos. So they should have or they should gain the property of binding with MHC. So in the cortex of the thymus, they'll get opportunity to bind with the MHC. So there will be cells that can able to bind with MHC and there will be cells that are unable to bind with the MHC. So we are selecting the cells that have this property, because this is the important property of T cells. So we are selecting those T cells that have the property to bind with MHC by directly picking up these cells. So this is a type of positive selection which is happening in the cortex of thymus. What is happening in the cortex of thymus? They are getting chance to interact with the MHC. So the cells that can interact with MHC will get selected, and the cells that cannot interact with MHC will be discarded. Now, once they interact with MHC and got selected from here, now these cells became double positive. That means now they contain CD4 plus also and CD8 plus also. CD4 and CD8 markers. Now they will enter the medulla of thymus. Now here they will undergo negative selection. How this negative selection takes place? Now the cell contains both the markers CD4 and CD8 passed from positive selection. Now again they'll get opportunity to interact with self cells. Now another property of T cell is T cell should only interact with the antigens or the foreign cells, not the cells of our own body, because the T cells, they interact with the cells that got infected or the cells that are foreign to our body. So they should not interact with our self cell. So in the medulla, they'll get opportunity to interact with self cell. But what they should do, they should not interact. If any T cells got in interaction with self cells, that means these are harmful to our body, because then T cell will start attacking our own cells only. So this is harmful to our body. Clear? So from here, these cells will be discarded, and that's how these cells will get selected. So this is a negative selection. So T cell, first of all, it should interact with MHC, and second, it should not interact with our self cell, then it will be harmful to our body, because it will start killing our own cells only. So they should not interact with our self cells. Clear? So that's how they will pass the negative selection. And after passing the negative selection, now when the cells are both DP, they'll get signals, and depending upon the signals, they will either become CD4 plus or CD8 plus. And now the cells are TH or TC cells ready to perform their function. Clear? So that's how the T cells undergo maturations and differentiation. So that's all for this video.